video comes to you in 8 parts. Part 1, the intro slide. Part 2, today's topic. Researchers often need to do experiments on machines that are too expensive for them to have one per lab and too complicated for everyone to know how to use them properly. So there are specialised facilities where permanent trained operators run and look after these machines, and researchers come from all sorts of different labs to use these machines in these facilities. Today we're going to look at some of these machines in the facilities and the people who manage them. Part 3. Part 4. Part 3. I'd like to introduce you to Hamish, who runs a large NMR facility for Melbourne University. Unfortunately, Hamish is sick at the moment, so how about I give you a brief tour instead. These are NMR spectrometers, and there are 8 in the facility here that Hamish runs. NMR spectrometers are used by chemists, biochemists, structural biologists and molecular biologists to identify the structure of molecules and proteins. The NMR spectrometer is mostly comprised of a large electromagnet. And the larger the electromagnet is, the stronger it is, and so the more sensitive the machine is. This one over here is a small, simple one, but this one over here, considerably stronger, much more sensitive. Perhaps understandably, machines this large and complicated are often quite expensive. But I don't know how much they are off the top of my head. Hamish trains students to use these machines on a regular basis, looks after the complicated machinery, troubleshoots the complicated software programming of the machines, and often aids researchers setting up more complicated experiments. So a researcher inserts their sample into the machine via that hole at the top. Most of these machines are operated manually. The one over here has an automated sampler on it. Let's take a look at the machine's robotic arm, taking a sample out of the machine and putting a new one in. Each of these tubes has about 2 ml of sample in it, the compound of interest dissolved in a liquid. Air pressure keeps the sample at the top and then sucks it into the machine when it's ready. I hope you get better soon, Hamish. This facility would probably fall apart without you. Many of these research facilities are located in different buildings around a whole lot of different research institutes and universities, and often researchers need to leave their building they're in to go and use these facilities. Which means we get to go outside! Part 4, Banter and the flow cytometry facility.
This is what's called a flow cytometer. It is used for researchers who have mixed populations of cells, but they want a pure population. We've got mixed populations. You're looking for a result. You don't know which of the cells is actually doing the job that you think is going on. So in this facility we've got 2 machines. One is the sorter, will identify the cells that they want on the screen and then get pure populations. We have this much smaller machine here, but this one will only identify the cells on the screen. I probably have at least 2 or 3 clients who come through each day. Some days I just have 1, some days I might have 5. Most of the people who come through here are from biological science. We run the samples for people, we make sure nothing goes wrong here. We also train people to learn to use the analyzer by themselves. The other things we do is analysis for people who don't know how to interpret these data. If they don't know what they're doing, we actually tell them how to extract their cells, how to label them up so that they're able to identify the cells that they're interested in. This particular one was bought about 3 years ago and it was around $500,000. This machine, just under $200,000. I did my PhD in blood stem cells. Decided that academia wasn't for me, and I decided to run this sort of facility instead.
For part 5, we're going to look at a facility that has 2 different types of machine in it, and the manager there is Nick.
We have 6 mass specs and 4 HPLC systems, and they're all used for identifying either proteins for purifying proteins. So a biochemist might have isolated a protein and then they would try and figure out what the identity of that protein is, and so they'll use the machine right there. Also the chemists come down and measure their molecules and go, did we synthesize what we thought we synthesized? My normal job is fixing all the machines. I take care of all the service contracts, and we have about 300 actual users. We teach them how to use all of these different machines. By having us teaching them, it means that we can actually have a lot higher throughput than what we would if it was just myself trying to run every experiment for them. I'd never be able to do half of what we get done in here in one day. Answering inquiries as to can our machines do this, can they do that, and I could explain that maybe they can't do this experiment but we could do something similar or something related that might give them the answer that they were after. About 300 users, probably at least 60% of them are from the University of Melbourne, which is local. But then we also have users from every other Victorian university. We've also had people from interstate. So we've even had someone come from England to use the facility. The machine beside us is about $100,000. The mass specs in the lab vary between $350,000 all the way up to about $1.2 million. I did my PhD in a lab, and we had one of the first mass spectrometers in Australia in that lab, running the machines all the time. That's what I was good at here in the lab. I took over in 2009. Somehow, miraculously, I'm still here.
Les doesn't operate a machine that researchers run experiments on, but he provides an equally useful service that aids researchers in their work. Les is Melbourne University's resident glassblower.
My main customer is chemistry, but I look after glass requirements of the whole university. Also look after RMIT, La Trobe. A lot of the work is done by hand because it's asymmetrical. So I use a torch here. Anything bigger, it's a glass lathe. Lathe different to a metal lathe, it's got two chucks, so that you can join a small piece onto a larger piece and it won't twist under if you're doing it. A lot of people come to me and they're not quite sure what they want because they're not quite sure what can be made in glass. And if you spend just a few minutes with those people, you can often send them away pretty happy with a nice design. Because people aren't used to realising what actually can be made in glass, and it's actually very versatile and wonderful material. All of the glass that you're seeing here, this is all borosilicate. The type of glass you get in your windows is a soda lime glass. Borosilicate has got a low expansion rate. That's why when I heated that tube there, it didn't crack. I heated it very quickly. Normal glass would have shattered. Okay? It's got good chemical resistance, and it's an ideal glass for the laboratory. And nearly all the glass in your lab is borosilicate. Because borosilicate has got a higher melting point, you've got to use more expensive refractories to make it. In my day, we did what was called a chemical diploma. As part of that course, we actually studied glass blowing. I've been working in glass ever since, and I've been working in research labs all my life.
And part 7, the bit where I go back to work. Les the glassblower showed me some amazingly cool things that I didn't have time to fit into this video, so if you want to see an explanation for this...
So what I did was I made the nail up out of window glass, and the mosquitoes will come and feed. The glass is very strong, you can hit it with a hammer and it won't break, but if you break the tail...
Check out this video in the link.